Okay, sir. Good morning, friends. Today I'm here to take class on COVID-19, the mass killer. So we all knew about COVID-19 in news and also I would like to give a brief idea about what is COVID-19 and how it is affecting our body. So the contents I include is definition, etiology, epidemiology, pathophysiology, signs and symptoms, diagnosis, and management. Before going on to this topic in further, there are few things that you have to know. One is why COVID-19 is known as novel corona. These are all basic things. So I wanted you to know all these definitions and meanings. So this is one is what is novel corona and what is series interval. And series interval and reproduction ratio, they are actually connected. So, and then positive sense single stranded RNA and ARDS, acute respiratory distress syndrome. So, I hope you will be able to answer all these questions once if I complete this template presentation. So, going on to the definition, it is named as SARS-CoV-2. It is named by the International Committee on Taxonomy of Virus. SARS means severe acute respiratory syndrome. Why is it known as SARS? When I discuss in the further slides, you will understand. WHO mentioned and termed this coronavirus as COVID-19 because this was first reported in the year 2019. So in short, they named it as COVID-19. So now I'll come to the term novel coronavirus. I have already asked you what is mean by novel coronavirus. Novel coronavirus means it is a new disease. In the history till now, we never have been reported any disease like coronavirus or COVID-19. It's a new disease. But if you check in the past 2000s, past decade or in the 2002 and all, there had been diseases of corona, but of same family, but different species, but of different genus. So novel disease means it is a disease that didn't exist, but the pathogen doesn't, means the pathogenic disease doesn't exist, but the disease from the same family is known before. Have you got what I'm telling? Hello?
Yes.
So okay. Okay. Novel disease means that disease does not exist as itself, means it has not been reported till now in the history. But but from the same family, some other disease have been known. But from the same family, there are some other disease have been known. That's why it is termed as novel coronavirus. So coronavirus disease is a potentially severe acute respiratory infection. So its procedure of affecting our human body starts from respiratory infection, and it is caused by severe acute respiratory syndrome coronavirus 2. That's why it is known as SARS-CoV-2. Why CoV-2 means, as I told you, it is all COVID-1 means COVID first disease have already been reported in the past decade. Coming to the etiology, we know that the etiology till now, research until now, people are trying to find out what is the etiology, but initially discovered in bronchoalveolar lavage samples in patients in Wuhan city, Hubei province in China in December. So SARS-CoV-2 belongs to the Sarbecovirus subgenus of the Coronavirinae family. Keep in your mind Coronavirinae family. I told you, right? As CoV-2, there was no disease till now, but from the Coronavirinae family, there was disease in the past past decade. And it's the seventh coronavirus known to infect humans. The virus has been found to be similar to severe acute respiratory syndrome like coronaviruses from bats, but it is distinct from SARS-CoV and Middle East respiratory syndrome. What is Middle East respiratory syndrome and what is SARS? We'll discuss in the epidemiology. Sorry, we will discuss here only. I'm sorry. See, can you see that officially named SARS-CoV-2, MERS-CoV, the officially named column 2019 nCoV, SARS-CoV-2, 2012 nCoV, MERS-CoV, MERS-CoV, HCOV, HKU1, 2005, and 2002 SARS-CoV. That means coronavirus was there, but not the coronavirus 2. Coronavirus 2 had happened in 2019. So there were diseases, Middle East respiratory syndrome. Why is it known as Middle East syndrome? Means initially it was in bats, then it got the virus got transferred to the camels. But you may be wondering then, like, we are every day we have to face one or the another animal or one or the other pets or something, but then why this virus is not getting transferred from the animal to the human beings? There comes the term recombination. There comes the term recombination. As the word suggests, recombination of the genus happen, of the genomic sequence happens. So what happens, this virus, especially coronavirus, I told you that is positively sensed single stranded mRNA. The genomic sequence is positively sensed single stranded mRNA. What is this? I want you to know what is this positively sensed single stranded mRNA. So I would like to stop this share 1 second. Whiteboard. Can you see a whiteboard now? Hello.
Yes.
Hello.
Yes, Sharon.
Yes.
Okay.
Whiteboard.
So yeah, this is the 5' end. You know that, right? 5' end and 3' end. Hello.
Yes, we hear.
Yes. And the 5' end is the cap. For the virus, this coronavirus, the 5' end is the cap and 3' is the tail. Since it comes, the cap comes at the 5' position and the tail comes at the 3' position, it is known as positively sensed. You got my point? It is known as positively sensed because at the cap 5' position is there and at the tail 3' position. If it was an inverted, we will say that negatively sensed. You understood? Now what happens? Okay. As in an RNA or mRNA or any genomic sequence, the three things in the genomic sequence is ribose sugar, phosphate moiety, and the nucleic acid base, right? Hello?
Yes, we hear you.
Okay, the ribose sugar actually is a 5-ring structure. 5-ring structure. This. It is a 5-ring structure. So what happens? Here is one H, OH, H, OH, H, okay, OH. Okay, here is one H. This is the structure of a ribose sugar. So what happens? This is the first carbon. This is the second carbon, third carbon, fourth carbon, and fifth carbon. So what happens? In 5' position, this can you see the fifth carbon? To the fifth carbon, that OH phosphate moiety will be free. And in the third position, one phosphate moiety will get attached. In the fifth carbon, hydroxyl group will be free, and to the third position, phosphate moiety will be happening. This is the 5' position. How we will determine the 5' position? See the fifth carbon. Can someone mute, like, my voice? Yeah.
What is that background voice?
So
now?
in the,
Who is causing that?
yeah.
Okay, go ahead.
Farshad, Muhammad. Okay. So 5' position, how we are going to detect? See the fifth carbon, hydroxyl group is there, free hydroxyl group is there, that will be the 5' position. And see the third carbon, if phosphate group is attached, that is the 5' position. How we will detect the 3' position? This is ulta. Sorry. The opposite will happen in case of 3' position, means here phosphate group will be attached Here phosphate group will be attached, and here hydroxyl group will be there. You got it? This is how we determine the 5' and 3' position. So this since the cap of the RNA is 5', and since the tail of the RNA is 3', it is known as positively sensed single stranded RNA. RNA means single stranded. Here it is only one strand is there, so positively sensed single stranded RNA. Now coming back, share stop here, share content, Google Drive. Okay. So what happens? I told you, right? This RNA, there is an adaptive ability for this viral RNA, viral genomic sequence. What is that? Recombination. Means if it gets any other RNA or DNA, what happens? It will recombinate with the other genomic sequence. So what happens? We cannot do anything at the molecular level. That's why till now for HIV, these medications have not been found, because always the continuous modification, continuous recombination is happening. Because of this continuous recombination, the viral genome from the bats are able to transfer to the other species. You got it? Hello? Hello?
Yes.
How this,
Yes.
hello?
Yes.
Yeah. Okay, how this transfer happens? The transfer happens because the viral genomic sequence has the property to go continuous modification. So because of this continuous modification, this virus is able to transfer from one species to the another species. So in 2012, it was transferred from bats to camels. Camels are seen in these Arabian nations and Middle East nations. So it is known by the name Middle East virus. That's all about that. Then coming to the epidemiology. This is a report from 31 December 2019. Okay, this is like I didn't get updated information, so I thought I will take this. So as you know, China was the first nation to be infected with coronavirus. And if you see, the majority of confirmed case, this is report from 31 December to 11 February, okay? So the majority of confirmed cases, 87% were aged from 30 to 79 years, and 1% aged from 9 or younger, and 1% were 10 to 19, and 3% were 80 years or older. And 51% of patients were male, and 49% were female. Nearly 4% of cases were in healthcare workers. That is, it is a nosocomial infection, means it can get transmitted from any hospital kind conditions. Nearly 80,000 cases and approximately 2,800 deaths have been reported in China. So the majority of cases are in Hubei province. Then these are at least 7,000 cases and 104 deaths. This reports, I mean, these cases have increased now, but still I'm just telling. As of March 2020, 50 countries outside China have been reported with coronavirus, and the names have been given. You can go through it. And to measure that, so, and I have asked about two terms, right? Reproductive ratio and serious interval at the first initial section. So I'm going to explain here what is reproductive ratio or series interval. Reproductive ratio means to a degree of spreadability. So what happens here? To how many person can one person infect? Can one person with coronavirus can infect? How many person it can infect? That is defined by the term reproductive ratio. And according to an estimate, one person having coronavirus can infect an average of 3 people. As of now, can infect an average of 3 people. So you should see, one person can infect 3 people. So these 3 people can infect another 3 people. So it will become 9. This 9 people can infect each person can infect another 3 people, so it gets transmitted to another 27 people. So the ratio goes on. It is an exponentially increasing graph if we see the reproductive ratio. Now coming to the series interval. So once again, coming to the series interval, consider that one patient has corona. This is patient number A. Okay, patient number A has corona, and now he developed symptoms. Sorry, he developed symptoms on the first day, and that, and he had stopped his symptoms in the 14th day. So this was like the incubation period. Okay, 1 day to 14th day. This is patient A, and this is patient B. Patient B. Now, just as this patient can infect the other patient in this duration, 1 to 14 days duration, the patient A can infect the patient B. Consider that he infected the patient B on the third day. On the third day, okay. So I'm drawing. It's like 14, okay. And this is the first day. Now, if you draw, this is called the series interval. This is called the series interval. That means the onset of symptoms of patient A to the onset of symptoms to the patient B. This is the series interval. This is the series interval. The onset of symptoms from patient A to the onset of symptoms to the patient B. That is known as the series interval. Now, if we see the reproductive ratio, I told 1 to 3, right? For coronavirus is 1 to 3. Reproductive ratio. And the series interval, it is said to be like 5 to 7 days. It is actually very short. 5 to 7 days. Within this time period, the chances of other people getting infected is really high. So it is known to be 5 to 7. So if you see this 1 to 3 and 5 to 7, both are high. I mean, this is comparatively low and this is comparatively high. So what happened? The rate of infection increases. In epidemiology of infectious disease, we see for these two things, reproductive ratio and series interval. If series interval is low, that means many people can get infected in a short span of time. And reproductive ratio is high. That means the number of people who can get infected are high. So when reproductive ratio is high and series interval is low, which is happening in case of corona, so that means the infection can spread in an unimaginary way. That is the meaning of this reproductive ratio and series interval. Ayo. Okay. Coming to the pathophysiology. Coronavirus. Curves are enveloped positive single-stranded RNA virus with nucleocapsid. I told you already what is meant by positively single-stranded RNA virus. Positively means the cap of the RNA was 5' and the end of the RNA was 3'. This is a general structure of a virus, not a COVID structure, okay? So you can see envelope protein is there, envelope is there, viral genome around that nucleocapsid to protect the viral genome, and viral tegment to give the structure for that virus. So this is the structure of a COVID-19. Curves are the largest known RNA virus with a 5' cap structure, 5' cap structure and 3' poly A tail. That means it is a positive stranded RNA. So when you discuss about this positively stranded RNA, you have to keep in mind certain things. That is certain things like what is negative stranded RNA. Then only that positive stranded RNA concept becomes clear. So a negative single stranded RNA means it does not cause infection as its own, Okay, so just like a carrier. It does not cause any infection. This negatively sent single-stranded RNA virus does not cause any infection. Once it gets transferred to the positively sent single-stranded RNA, then the infection can proceed. Infection can be made by that virus. So what happens here? This in this coronavirus, it is positive sense, and this positive sense single-stranded RNA can work as messenger RNA. Why it is infectious means positive single-stranded RNA can work as mRNA. It is not a different thing. By hearing this messenger mRNA or the messenger RNA, don't think that this positively single-stranded RNA got transferred to messenger RNA. It is not like that. It is also called as messenger RNA because it is positively stranded. So it can act as messenger RNA. So it can be seen in hepativirus C, then dengue virus, SARS, MERS, and in even this COVID, rhinovirus. So all these things, all these virus has this positively single-stranded RNA. So if you see the structure of this RNA, structural protein, it is, can you see that envelope protein in the green color circle? That is called the envelope protein. What is its duty? Interacts with membrane protein to form viral envelope. Envelope protein interacts with membrane protein. Can you see some light green structures embedded in the envelope protein? So this envelope protein and membrane protein together form the envelope of the virus. So inside that there is RNA and the nucleocapsid protein. Inside the virus there is RNA and nucleocapsid protein. This nucleocapsid protein binds to the RNA genome to form nucleocapsid. That's what we see in here. I told you, right? Nucleocapsid structure. Can you see? Hello?
Yes. See, all of you have to respond, no? Going a very interesting session.
Okay, sir.
Yes, sir.
Okay. Then now let me stop here about, and then one more thing, this spike protein. Spike protein is, what is this spike protein? Spike protein, it is made up of spike glycoprotein. Spike protein is made up of spike glycoprotein, and critical for binding of host cell receptors to facilitate entry of host cell. So I will discuss that in later. What is the spike protein? How is it important? And what is its role? I'll discuss in the pathophysiology. So coming to the routes of transmission, the main two routes are fecal oral route and airborne way. That means fecal oral route means someone going to washroom and they won't wash their hands properly and they will touch taps, wash basins. And in a short duration of time, within a span of 3 hours, someone coming and touching the surfaces again. Then what happens? Then the other person, there is a high chance of getting infected. That is with the fecal oral route. And the airborne way. Airborne way, as you know, I have already, like while talking about tuberculosis, I have mentioned one term like droplet nuclei, aerosol. So in airborne way, these aerosols, like with the virus carrying our droplets, virus carrying droplets will come out when we sneeze or when we cough, and the other person who inhales it, he gets the disease. That is what is meant by airborne way. And why I gave this structure of lungs, you have to see what is pleural cavity. Can you see an ash color structure? That is the pleural cavity. And now coming to what are alveoli. Alveoli, it is the end point of the respiratory system. Alveoli is the end point of the respiratory system. And exchange of oxygen and carbon dioxide in the bloodstream. So it's not only this exchange of oxygen, gaseous exchange is not the only function of alveoli. There are many other functions, but the main function can be exchange of oxygen and carbon dioxide. But you should remember, alveoli is the end point of the respiratory system. So type of alveolar cells. Once we discuss about lengths, then coming to the alveoli, the types of alveolar cells is pneumocytes type 1 and pneumocytes type 2 and alveolar macrophage. These are the cells which are present in the alveoli. If you see pneumocytes type 1, that is responsible for the gaseous exchange. Pneumocytes type 2, responsible for production of surfactant. What is the function of surfactant? Can anyone tell? Function
To
of
reduce
surfactant?
surface.
Yeah, to reduce the surface. So then alveolar macrophage. How this surfactant? The surfactant is the main hero of this spreading disease. So we will see how is it affected. So alveolar macrophage is mechanical defensive respiratory system. We know that macrophages, what it will go and do when any foreign bodies enter our body, it will go and eat that foreign body. That's why it is called as mechanical defense of respiratory airway, because it is alveolar, part of the respiratory system. That's why respiratory airway. Now, this is one picture that I drew. This S-spike glycoproteins I have mentioned. Can you see the virus structure at the top? Hello?
Yes.
Yeah.
Yes, we
Yeah.
can see.
S-spike proteins, this virus molecules enters into the alveoli. This virus molecules enters into the alveoli, and you can see it is going, and in the alveoli you can see surfactant producing cell that is given in orange color. And then you can see below the alveoli there is the interstitial space written in brown sketch, interstitial space. Below that there is one tube-like structure that is blood capillary. So I have redrawn this surfactant cell again. This surfactant cell, okay, it is pneumocyte type 2 cell responsible for the production of surfactant. Surfactant reduces the surface tension. Reducing the surface tension means, so inside the alveoli, when any pressure comes, so it reduces that tension. If pressure becomes too much, what will happen? This alveoli will not be able to withstand that pressure, and collapse of the alveoli will happen. So this surfactant, when I redrew, can you see a pink color structure? That is the receptor present in the surfactant. Its name is ACE2 receptor, angiotensin converting enzyme type 2 receptor. So what does it do? Now comes the role of S-spike glycoproteins or spike proteins present in the virus. This S-spike glycoproteins has a particular domain which has high affection towards the ACE2 receptors. These ACE2 receptors are in turn can be seen in GI, in the duodenum, in the intestine. That's why we now the government has said, right, diarrhea instead of fever. They are telling diarrhea is one of the main symptom of corona, because these ACE2 receptors can be seen even in the GI tract. Now, talking about the lengths, in the surfactant cell, ACE2 receptors are present. So this spike proteins has a domain which has high affinity towards the ACE2 receptor, which in turn allows the virus molecule to be engulfed in. So once it is engulfed, it releases the genomic sequence, that is the mRNA. The genomic sequence, can also work as mRNA. It can be said as mRNA because it is positively sensed single-stranded RNA. So what happens? This positively sensed single-stranded RNA, once it is released, the ribosome is present. Ribosomes are present in the surfactant cell. So once it is released, once the genomic sequence of the virus is released, the ribosome, this mRNA, or it can work as mRNA because it is positively sensed. So this mRNA will go to the ribosome. What is the duty of ribosome? Synthesis of proteins. So once it gets the mRNA, it begins to